Hello guys, welcome to my YouTube channel at Need Biology. So now I'm here with you with very important points from NCRT from a chapter called Biotechnology Principles and Processes. Okay children. So the most important point here is first is tools for recombinant DNA technology. So as you can see here, what happens in recombinant DNA technology? What do we do in recombinant DNA technology? So children here, desired DNA from any cell, we take out the desired DNA and that is introduced into the host cell, but we cannot introduce it directly into the host cell. So first we have to extract the DNA, cut the DNA from any cell. That is done by using enzyme called nuclease, restriction endonuclease, and then to join we need an enzyme called ligase. Okay, and that DNA is introduced into a vector and then introduced into host cell. Then comes principles of biotechnology. In NCRT, you are studying two principles. One is genetic engineering and second is bioprocess engineering. Then coming to, there are some important questions asked from the first recombinant DNA. Okay children, that is, it was given by Stanley Cohen and Herbert Boyer. Next, which vector was used? Plasmid of Salmonella typhimurium. And which was the host used here? That is E. coli. Okay. Next, coming to restriction enzymes. So, more than 900 restrictions enzymes have been discovered from more than 230 bacterial strains. So, this information is given in NCRT. It is very important. Then coming to first restriction endonuclease, that is HIND2. Okay. Then the vector used for plants is Agrobacterium tumefaciens and vector used for animals is retrovirus. Okay children. Next coming to, yes, as you can see here, types of nucleases, activity of nucleases. So there are two types of nucleases, endonuclease and exonuclease. The DNA strand, endonuclease means it cuts the DNA at the exactly like the center or internal cuts is called endonuclease. And if the DNA is cut from the ends is called exonuclease. Next coming to gel electrophoresis for DNA. Gel, what is the gel used here is agarose gel. Next, what is the meaning of electrophoresis? Electrophoresis means movement under electric field for DNA. Next, to see or to visualize the DNA, stain with ethidium bromide under UV light. Okay. Next, now there are two more techniques here. One is microinjection, other one is biolistics or gene gun method. But don't get confused. How to remember which method is used for plants and animals? See, you remember injection. Usually injection is given to animals or humans, you can say. So microinjection for animals and biolistics or gene gun for plants. Okay. Next, to break the cell, to break the DNA, if you extract the DNA, we have to break the cell wall of different organisms. So, to break bacterial cell wall, we need enzymes called lysozyme. And for plant cell wall, we require cellulase. And for fungi, we require chitinase enzyme. Okay. Next one. Bioreactors. There are two types of bioreactors. Simple stirred tank bioreactor and sparked stirred tank bioreactor. Simple, as the name indicates simple, here no formation of bubbles. And here sparked stirred tank, here bubbles are formed. And the last step is downstream processing. It is the most important children. Downstream processing includes two steps, that is separation plus purification. Now how to remember this? In chemistry you are studying periodic table, right? D block, S block, P block elements. So D block is equal to S plus P, separation and purification. I hope you understood the important points about this chapter. Okay, thank you.